So for the first talk, uh, it's going to be by Jan Skotheim from the Stanford University. It's about the modular organization of eukaryotic cell growth. And so without much further ado, I'll let you.
First, I'd like to thank the organizers for the invitation to come here. Kind of like Uri Alan this morning, I probably worked most closely related to the interests of many of you at the conference here about 15 years ago, when we were looking at kind of the modular organization of the cell cycle and signaling pathways that regulate the cell cycle. I guess I'm happy to see that those ideas that we were talking about are still very relevant and alive in the community here today. And so it's been also interesting for me to see, the kind of aspects of modularity that this group is worried about. I mean, haven't been to a conference like this before. So since I would say over the last 10 years, my work has kind of shifted from some more of those like signaling pathway ideas to more looking at quantitative aspects of cell physiology. And that's what I'm going to be talking about today, about the things that we've learned by studying cell growth in a quantitative way. And I'm kind of hoping at the end of it, there's a slide at the end of it meant to kind of connect to the different topics here. So I guess bear with the quantitative physiology, and then hopefully we'll get back to some of the things that are, I don't know, maybe more topical for the group. Right. So the main question about cell physiology that my group has been working on is this question of how it is that cell growth triggers cell division. So it's known in a lot of different contexts that cells grow, and this fact that they get larger triggers a signaling pathway that drive cell into the cell division cycle and make them become two cells. And so the question that we became really obsessed with is this question of how does it this works, right? So what is it that's really different about this cell that's larger, otherwise looks kind of the same as the smaller one? What is it that's biochemically different about that cell that makes it trigger cell division? And so when we started looking at this about 20 years ago, I would say we didn't know how this worked for any type of cell, much less how this could be modulated to hit, over 1 million-fold range in target cell sizes that exist in the human body. And so our approach was to address this with the simplest organism that we could think of trying to address it in, which is also related to eukaryotes like us, and that is the model organism budding yeast, which is amazing for many different reasons, but that's the organism that we decided to use. And so we made some progress, right? And so to review 20 years of progress, it looks something like this. I would say our main discovery in this area was that growth could act to dilute out inhibitors of cell division. It was a bit counterintuitive. When we started looking in this field, we and everybody else were thinking that cells get larger, there should be more of some kind of activating molecule that triggers division. But the cell doesn't really care what our intuition is and does it the way that it wants to do it, or it evolved to do it. And that way is through this dilution of inhibitors of cell division. So in budding yeast, there's a protein called We5, which is diluted by cell growth. Following this discovery in budding yeast, we moved into human cells, and we discovered that the retinoblastoma protein, which functionally does something similar to the We5 protein, and that inhibits the transcriptional program associated with the cell division cycle. Evolutionarily speaking, they're completely different beasts that don't look like each other at all in amino acid sequence, but functionally they're both inhibitors of cell division, and they both exhibit the same kind of size-dependent concentration dynamics that connect growth and division in the cell cycle. So following our discoveries in yeast and human cells, a couple more reports came out indicating that this general mechanism also takes place in plant cells, in Arabidopsis, and in alga, in Chlamydomonas. But the identity of the inhibitor of cell division, which is diluted in all cases, is totally different. So there's kind of a principle here that reappears or re-evolves to link growth and division in a variety of different types of cells. Okay, So that was like a really foundational discovery for my group. That was 2015 paper. It was the first one where we reported the dynamics of We5. And since then, we've been moving in 4 different directions. Yes?
So if you take cells that are not growing and you just somehow reduce the expression of these inhibitors, does that result in cell division without growth?
That's a complex question, actually. So sometimes.
Okay.
So if I was going to go back, so what I'm talking about here, we tried to study this in the simplest way that we could, which was a constant environmental condition. Nothing is changing. The cells are, biomass is kind of exponentially accumulating, and we're not reducing the growth of the cell. When you reduce the growth rate, actually, like as you suggest, that impacts the concentration of the activators. So in a way, like this activator signal is the growth rate.
Yeah.
And then this dilution is the cell size. And both of those signals independently can regulate the transition. Yeah. Great question. Okay, so the 4 directions we've been looking at, one is kind of to just characterize more generally what's happening in terms of how the cell composition changes as they grow larger. And that's been in a lot of quantitative proteomics. Secondly, we're very interested in like what are the, having now found that We5 is diluted, what are the kind of molecular apparatus that the cell uses to generate that really useful dynamic pattern, also for RB. Thirdly, Mimi Shi, who's a postdoc in the group, is also here at the conference, had a poster. We're interested in looking at how these kind of cell autonomous mechanisms, such as this inhibitor dilution type mechanism, function in the context of a multicellular system. So she has a poster here, so if you're interested in that. And then finally, like the more we've been thinking about how to couple growth to division, we start to think more and more about just really like how do cells grow, just like the growth part of the question. And this is what we're going to talk about today. So, we start off, get excited about this question of growth linking to the cell cycle and how that works, but let's forget about the cell cycle part. Let's just think about the growth part. And I think, as you'll see, that's quite an interesting question itself. So we're going to think about it in 2 different contexts. One is we're going to think about it as cells get larger. So in like a specific growth environment, the cells grow and get bigger and bigger and bigger. And what are the kind of problems that the cell might encounter while it's doing that, And secondly, we're going to think about what happens as the cell grows in different environments, as it grows at different rates. And how does the cellular like kind of composition organize change to reflect that? Okay, so let's have a look at the specific condition. So as cells grow larger, the cell size increases. Most of the things in the cell also increase in proportion to size. So like if you look at something like the nucleus or mitochondria, volumes, vacuole, these things all tend to kind of proportionally or close to proportionally increase in size. Macromolecular constituents like RNA proportionally increases, protein, et cetera. These things also increase. And this makes some sense that you might want to try to keep roughly your concentrations relatively constant as you're getting larger and larger, because this will help you keep your biochemical reaction rates where you presumably want to have them. Right? So this is the idea of maintaining roughly similar composition. Right. Out of all of those steps, I think the least trivial one is to think about how do you scale up the amount of RNA with the size of the cell, right? Now, why is that? Well, it's been noted that the synthesis rate of RNA increases with cell size, and this is true even in cells where you have a fixed amount of DNA, right? So you have a fixed genome, or if you're going through the cell cycle, at some point you have a discontinuous increase in the amount of DNA in the cell, yet you get this very smooth accumulation of of mRNA, right, to maintain it at constant concentration. So this is confusing, right? How does that single genome know that the cell got larger and knows to then, upregulate the synthesis of mRNA? So this is the first question that we're going to try to address, right? So how do larger cells transcribe more than smaller cells? So the key quantitative experiment that Matthew, who was postdoc in the group, did to try to measure kind of global transcription is this spike in normalized chromatin immunoprecipitation experiment. And he uses this spike in order to get not just relative distributions of polymerase across the genome, but really an estimate of the total amount of polymerase on a genome, right? So it's not just like relatively where is it, but really total amount of it on the genome. So that's what the spike in gets you. And so we can take sort of different mutants that we've generated where they have different cell volumes, and then we can do this experiment, and you can see that the bigger the cell is, the more RNA polymerase II you have on the genome, as you would expect because you're transcribing more. And that's true for like different versions of RNA polymerase II. It doesn't really, these details don't really matter too much, right? So why does a cell load more RNA polymerase II on the genome? So one possibility, I'm in a biology department, so I realize like, the chromatin should be explaining all this stuff. And so one possibility is that the chromatin, something changes about the chromatin as the cell gets bigger, maybe it gets like looser, more open, and then that makes it more permissive for the polymerase to load on, so higher kind of on rate. We did some experiments to test that, and I don't want to get too bogged down in the details other than to say that kind of independent of the size of the cell, these curves really look identical. So any kind of measurement we made of chromatin accessibility doesn't really change as the cells get larger. So that's not that, right? So chromatin accessibility is not it. We're still stuck with how does more Pol2 loaded onto the genome. Okay, so one hypothesis that had been proposed in this series of papers in the literature is that there's some kind of limiting factor, right? So there's some kind of limiting factor whose amount accumulates in proportion to the size of the cell. So this is just some protein whose concentration doesn't change. And that limiting factor, binds the genome and then activates transcription in proportion to the amount of this factor bound to the genome. And of course, the amount of the factor is proportional to the size of the cell. This is this kind of limiting factor hypothesis. However, it hadn't been identified what it would be or if this was actually true. Yes?
Just to clarify,
yeah.
Is nucleus size proportional to cell size?
Yeah, roughly.
Okay.
Yeah. So all of this stuff is going on in the nucleus, who's about, in budding yeast, growing in these conditions, is about 6% of the volume of the cell. Okay, so what could be the limiting factor? Well, we look at all of these different things that are essential for transcription, and they're all kind of at constant concentration, so their amounts accumulate linearly with the cell volume. So they're all potential factors, right? And they all, there are other measurements where we look at how much of it is associated with the chromatin, and they all accumulate in the chromatin as the cells get larger. All right. So let's test, these factors as potential models. So how do we want to do that? Well, we do it where we use this rapamycin-inducible binding system to anchor half of the particular protein into the cytoplasm, away from the nucleus, where presumably it's working on transcription. So we do this we have a heterozygous diploid where one allele has this FRB rapamycin-induced binding domain and the other allele doesn't. So we add rapamycin, and half of it goes out of the nucleus in about 10 minutes. And so we do this with all of these different subunits, and we can see the occupancy on the gene bodies, interestingly, don't change very much, which is kind of interesting, because all of these factors, these TF2 whatevers, they're all essential for transcription. So if you delete them, you're dead. But if you remove 50% of them, in most of the cases, you get like nothing, or you just get like maybe a few percent change, maybe 5% change in the amount of RNA polymerase II loaded. with one notable exception, right? So we have this one notable exception here, where this is a subunit of RNA polymerase II. So when you remove half of the RNA polymerase II into the cytoplasm, you deplete about half of the RNA polymerase II from the genome. So what does that mean? Well, maybe RNA polymerase II is the kind of key, limiting factor regulating the rate of transcription in the cell. All right, so let's test that. So one way to test it is not removing half, but trying to increase the amount of RNA polymerase II. This is pretty hard because it has 12 subunits. And so we created a yeast strain where all 12 subunits of RNA polymerase II are overexpressed by about twofold. And so we did that, and we got a little bit more RNA polymerase II on the genome. There's a lot of reasons to believe that we didn't get twice the amount of RNA polymerase II because there's a lot of like cofactors and chaperones and whatnot. But we got more, which is consistent with our model. Okay, so let's get a little bit more quantitative and make a real mathematical model out of this. And so to get more quantitative, we want to just control the size of the cell and the amount of genome. And so what we do is we mechanically isolate small cells that are in the big early phase of the cell cycle, G1 phase. We do that with centrifugation, basically, to separate out the small early G1 cells. Then we prevent them genetically from going into the cell cycle and dividing, and then we grow them. So they just get bigger and bigger and bigger. And so you can see these are the distribution of sizes of the cells as they're growing larger. And then for each one of these groups of cells, we can then measure the RNA polymerase II occupancy on the genome as the cells get larger and larger. And this is the curve that you get. You get this kind of like initially linear increase and then followed by a saturation. All right, so does the simple, titration model fit? Well, kind of. I mean, initially, yes, maybe. But then you have a problem here. So just a simple, linear limiting factor model doesn't quite work. There was later a proposal that there'd be a saturation step. So you have, titration, but then eventually you complete the occupancy of the genome. So you can't load any more polymerase. And so you would expect to have something like this. We have another model that we want to introduce, which is this, what we call it like a dynamic equilibrium model. And so here we have not just, all of the limiting factor bound, but, the RNA polymerase can float around the nucleoplasm. And then there's an on rate associated with it finding a suitable place on the genome, initiating transcription, and then an off rate with it associated with it falling off, right? And so then that's this equation. And then the second equation is just that it's at constant concentration so that the number of the RNA polymerase II is proportional to the size of the cell. And so you can make a model for that. You have, and we know that the growth of the cell is, I would say, slow relative to the transcription time scale of minutes. You're talking about hours versus minutes. So we can assume that this is in equilibrium. And then you can basically solve this, and you have essentially two parameters. One is the KD of the pol2 in the genome, and the second is the concentration of the RNA polymerase II. And so you have a two-parameter fit, which you can then fit the data, and so that works at least as well as the other ones. But the model makes very different predictions in terms of the fraction of the RNA polymerase II which is bound for any particular cell volume, right? So the titration saturation is fully bound till you saturate, and then it goes down. Whereas the dynamic equilibrium model is kind of like always, kind of like sloping downward as less and less of the fraction of RNA polymerase II is bound. So can we measure this? And so we sought to measure this, and so we did this with single molecule imaging. So we have single dye-labeled RNA polymerase II molecules floating around the nucleus. And so if they're bound to the genome, they don't diffuse very much, and if they're freely diffusing, they diffuse more. And so these are actual traces of the RNA polymerase II. These are traces of histones, so you can see the histones are all bound. And this is just a freely diffusing fluorescent protein, which is just flying all over the place, right? So you can kind of see here that some are bound and some are not. And so then we can measure that as a function of cell size, and you can see this fits pretty well with the two-parameter dynamic equilibrium model and is not consistent with this titration saturation model at all. So that was exciting. So how else could we test this model? Well, one other thing in this model that or I should mention here that this isn't fit, right? So we only fit the previous curve. This is just a pure prediction of our two-parameter model. Okay, what else could we predict? Well, we can change the amount of DNA in a cell by looking not only at previously we've just been looking at haploid cells, but we could look at diploid cells. And what does that mean? That just means putting the number 2 in front here, right? So again, we have no more parameters to fit. We've just put the number 2 there. And so then we can just make a prediction, right? So here's our haploid fit again, and this is our fit for the diploid. Right? So then we went and isolated, the same kind of populations of different sized diploid cells, and then measured the RNA polymerase II occupancy in the genome. And it was, I guess, over Christmas 2022, where we received this data, which was a pretty good Christmas present because it fit really well our model. So we're extremely happy to have our model make this prediction, actually. Okay, so first summary of this first part, we have this dynamic equilibrium model. If we take that, fully seriously, this is that we get this RNA polymerase II prediction, how much occupancy there is as a function of cell volume. However, if we assume, as had been assumed in the literature, that the mRNA decay rate would be independent of the volume of the cell, this isn't quite linear, right? So this is like it goes up, and then you have this saturation. It's not exactly saturation, but you have this plateau, let's call it. Because of that, because it's sublinear, what you would predict from this kind of calculation is that the mRNA concentration should go down and down, like the bigger the cell gets. So what would you need for the concentration to stay the same? Well, you would need the decay rate to decrease. The decay rate can't be flat. The decay rate would have to decrease. And so then we thought, well, let's measure the decay rate as a function of the size. And so we did that first we used a qPCR on small cells and then large cells. And so we can see the decay rate of this gene MET3 is a bit slower in the large cells. So we can estimate the exponential decay as a function of cell size, and we can see that it gets smaller and smaller. But not only that, it gets smaller in the amount needed in order to maintain the constant concentration, right? So that would be the model prediction. It's like if the concentration is maintained, this is the relationship that they should have. This is one gene MET3, but we can also see it with MET17, GAL1, GAL7, GAL10. So we did this for 5 genes. And of course, since you submit things to fancy journals that make you do horrible experiments. And so we had to do this experiment on all genes because 5 wasn't enough. And so we did that because we're motivated to, postdocs motivated to get jobs and stuff. And so we did that, and we did it for all genes, and it really looks, I think, pretty good. So this is the dashed line is what is needed to maintain constant concentration, and this is what we see. And so it's just a general global shift of all of the half-life. So we have gene level information here for every gene, and so it's just the whole curve is just shifted over. Right. So now we can see how you get constant concentration. It's pretty involved, actually. Like first, you have kind of a simple, elegant, I would say, model of mass action kinetics of the Pol2 in the genome, producing a sublinear increase in the transcription. And then to really get the full kind of maintenance of the concentration, you couple that with some kind of feedback control system, which we don't yet understand, where that control feeds back on the half-life of the mRNA, so that as you don't quite make enough, you then stabilize that mRNA so that to maintain the constant concentration. Okay, so those were the things we learned about this scaling up of cell size as cells grow larger in a particular condition, right? And now we're going to have a look on, how cell composition changes as we go into sort of different growth conditions. All right. So here, I would say we were very much inspired by the work looking at bacteria physiology and bacteria growth physiology. So particular, just highlight the work of Terry Watt at UCSD over the last couple decades. Has really done a lot to kind of like modernize the study of bacteria growth physiology. And we really like those models. Actually, I was on the hiring committee where we hired Jonas Kramer, who is a postdoc of, of Terry Watt. And, so I got to have discussions about these kind of ideas a lot more intimately. And that really inspired us to try to build these same kind of models in yeast and eukaryotic physiology to try to catch up to the prokaryotic understanding. Okay, so what kind of models are we talking about? Here's a recent example from Jonas, his first paper is published as an assistant professor, so it's like, go Jonas. Anyway, so in these types of models, they're simple, right? But they're not too simple. So each one of these arrows is some parameter in an equation. And so you typically have things like you have a metabolic sector of proteins, which converts nutrients into precursors, like charged mRNA, which are then converted by ribosomes into protein synthesis, right? And so here, there's an important coupling between all of these things, which take place because of essentially mass conservation. So the proteins that you make can be devoted to either the metabolic sector or the translational sector, or some sector of other proteins that you kind of need in order for housekeeping purposes of the cell, right? And so these models, these kind of compositional models are I think, interesting to me for many reasons. Like just as something you can study, it's great because you understand what all the parameters are. Like these are ribosomes and these are metabolic proteins, right? So you have a legible understanding of what all the equations are. You also know what the different, the 4 or 5 different equations are doing. One is making proteins, one is making, this. So it's like, it's a really legible model. And the number of equations and parameters are such that you can really go to town and analyze it in a comprehensive way. Okay, so that's all good. What can you get out of it? Well, you can actually make, with a few assumptions here and there, you can actually make some non-trivial predictions. So under some assumptions of, optimality of growth, like you're trying to tune the amount of ribosomal proteins for a particular metabolic condition, so you're optimizing the growth rate of the cell. In the end of the day, that means you have X number of metabolic proteins and et cetera, et cetera. You can predict things like the translation speed in amino acids per second of a ribosome moving along an mRNA out of these models, and then you can go and measure. And this is a really non-trivial prediction that fits, with this assumption of optimality will fit the data, whereas like other kind of assumptions don't. So it tells you, I think, some really non-trivial things about the organization of the cell to maintain, in this case, bacteria is trying to grow as fast as it can. All right, so let's have a look at some of these things for yeast. Let's look at how the composition changes as a function of the growth rate. So what do? Well, we just grow yeast in like 15 different conditions and measure the fraction of the proteome, which is ribosomes, and you get this linear increase in the fraction proteome that's ribosomes as a function of growth rate. This is true in our data as well as the previously published data. But how many of these ribosomes are translating? Again, this is an important number if we want to measure something like amino acids per second that the translating ribosome moves at. And so previously, there'd been a couple of reports trying to do this with polysome profiling. There's a lot of reasons why those aren't so quantitative. I won't get into it. And then kind of motivated by our, exciting work on the previous paper, being able to measure diffusion of single molecules, we thought, why don't we try to do this with ribosomes too, using the fact that mRNA is just massive, and so an mRNA-bound ribosome is going to diffuse slower than a free ribosome. And so we can try to do that, and again, the goal is to try to measure things like amino acids per second. So this is the previously published data on that, And I hope you can appreciate that there's not, like, two things. One, there's not that much of it, and two, it doesn't really agree with itself very well. So we're going to leave that aside and generate our own data. And so we're going to do that using just what I said. We're going to use single molecule tracking of ribosomes. Here's a movie. I think is the movie playing? Yeah, the movie is playing. This is what the data look like. So you can see these bright dots when, it's like translating, and then it's like diffuse and moving around when it's not. It's kind of cool. Probably like the Sky Institute could study it. No, anyway. But so, and the tracks look like this. And so we can measure the diffusion, the constant for these tracks, and they're kind of what you would predict based on the molecular weight, what you would calculate and predict based on the molecular weight. And we can fit these data with this two Gaussian mixed distribution. And then we can do controls, like we can here treat with this initiation inhibitor. So when we treat with LMT, what happens is that the ribosomes that have already initiated translation will finish and run off the mRNA, whereas those that have just bound are stuck at the initiation site. And so then you get this situation where each mRNA has like one ribosome bound to it and the other ones are free. And then we know that the ratio of ribosomes to mRNA is about 3:1, and that's exactly what we get. We get sort of 1/3 of the ribosomes are diffusing slowly, and 2/3 are diffusing quickly, as we would predict. So this is a good control for the model. And so these are the data for all of these dozen different growth conditions. And so for each one of these conditions, we can now measure the fraction of ribosomes that are active using our single molecule studies. And that looks like this. So as you grow faster and faster, you have more and more active ribosomes. Interestingly, you have this kind of like relatively constant fraction of inactive ribosomes, which can be quite a lot when you're growing slowly. So if you're trying to eat trehalose, for example, and dividing every 6 hours, about 10% of your proteome are these ribosomes that aren't translating. Lazy ribosomes. So the diffusion doesn't change that much in the growth rate. A lot of people are really interested in these physical properties of the cytoplasm, so let's put that up there for them. And so from this, now that we know the number of active ribosomes and the total protein synthesis rate, we can divide a couple numbers and we can get the elongation rate in terms of the average amino acids per second that these ribosomes translate as a function of growth rate. And it's pretty flat. It's about 9 amino acids a second. So that was kind of exciting for a couple of reasons. One is that it's very different than what was seen in bacteria. So it's clearly a different organizational system. The other thing that we can see, we can measure this as a separate measurement. We can measure the total mRNA concentration as a function of the growth rate. And we can also see a linear increase in the concentration of mRNA as a function of the growth rate. We can measure the concentration of RNA polymerase II. And kind of in the interest of time, I'm going to go a little bit quickly through this. But we're going to take, basically what we do is we take the same model that we had in the first part of the talk, and now we measure the nuclear volume. We measure the concentration of RNA polymerase II. We measure all of the different pieces of this dynamic equilibrium model to predict the mRNA concentration. And then, long story short, that works pretty well, right? So we can predict the bound fraction of RNA polymerase II, and we can predict the concentration of mRNA as a function of the growth rate. Again, with using our same dynamic equilibrium model that I introduced earlier, except now we independently measure the different parameters for all of these 4 different conditions. Okay, so what about the ribosome mRNA, right? So we know that the ribosome concentration increases with the growth rate as such. We know that the mRNA concentration increases with the growth rate as such. Can we put these two together to understand the growth rate? And so here we make another mass action kinetic model of like a free ribosome diffusing around, finding an mRNA, then translating that mRNA at a constant rate of 9 amino acids a second, and then falling off. And then out of these two things, we can then predict the protein synthesis rate. And so that works just fine. So you can see that prediction and reality worked pretty well. And the kind of interesting thing here is that there are lots of different ways that this model you could scale up growth. Like you could make more ribosomes, you could make more mRNA, whatever you do, because of the, they're both two parts of this mass action kinetics. They'll both increase the growth rate. The cell, for some reason, decides that it wants to proportionally increase both of them at the same time, right? So that's kind of what you can see in this plot here. I mean, there's some discussion about that. Okay, so this is just, I want to leave some time for questions, so I'm going to go a little bit fast through these tests. In one, we reduce the mRNA concentration to see if we can predict the growth rate. And in fact, we can. And this is a natural, and this is another experiment where we acutely, this one is kind of nice, you acutely inhibit the degradation of mRNA. So that leads then to an increase in the concentration of mRNA in the cell over shorter periods of time as such. And then that initially leads to an increase in the growth rate in the cell. About 15 minutes later, all hell breaks loose and the cell has a really bad time. But for 15 glorious minutes, it's growing faster than it's supposed to, and it's growing faster in the way that we would predict from our model. So this is the conclusion of the second part of the talk. We're sort of starting to catch up, I think, to the bacteriophysiologists and trying to understand, what kind of model is supporting yeast growth or eukaryotic growth. And, somehow it knows how fast it should grow based on whatever it's eating. Totally confusing, but somehow it knows this. And then it proportionally increases both ribosomes and mRNA. This then, through mass action kinetics, determines the number of active ribosomes in the cell. There's a constant elongation rate, which is totally independent of whatever it's eating or whatever it's doing. And then these two things together determine the total protein synthesis rate and the growth rate of the cell. So this is the current model that we have. So I hope you'll agree that we made, I feel like, a decent amount of progress in trying to understand different aspects of cell growth. Like in the first part, we tried to understand how cells scale all the different aspects of protein synthesis as they get larger in order to continue to grow efficiently as they get larger. And then in the second part, we're trying to build up this model for how the cellular composition is controlled and changed in an efficient way to control growth across different nutrient conditions. All right, so in the last minute, I wanted to, how does this maybe relate to the meeting? Okay, so modularity of growth. Is growth modular, or how might we think about growth as a modular process? So there's one part of it which is clearly not modular, and that is this comes through the kind of conservation of mass, right? Like you have to make one thing. If you devote some amount of resources to some module, then that means you have less to devote to the other. So there's really a kind of global coupling that comes through conservation of mass, which you might not think about if you're trying to think about like some signaling pathway versus another one, because the number of proteins that you're devoting to some signaling pathway or some signaling process is trivial. However, the amount of proteins that you're devoting to ribosomes is not trivial, right? So that's a different thing. Secondly, I think we ignore a lot of details. As you can see, like we're trying to understand aspects of growth, and we end up with like a 2-parameter model, right? So that this is where this comes in a way out of my training in applied math and soft matter physics, where you're trying to make the simplest possible model that you can make. Does the fact that this works? I'm going to say, okay, at this point, I'm convinced that this is a useful way of looking at it. Look, we make predictions, like we test them. Some of them even come true. And so clearly this is working in a way. What are the implications for, many aspects of the underlying system have to be more complex, right? Like what are the implications for the fact that these simple models work for what is going kind of like on underneath the hood? Does that imply that these different processes regulating this or that are modular or not? I don't know. But I think this is a question kind of raised in a way by the success of the simple model. Like why does the simple model work out? And then finally, I want to highlight a couple of things here. Like one is that all of the kind of models that I presented are really falsifiable. Like we can make tests, we can, prove them wrong. And the fact that we can do that comes from like the massive amount of effort that we put into generating like the best possible quantitative data, right? So it's not just like lots of messy genomics, it's highly quantitative data, which we're really convinced about it. It's convinced about it enough that, if the numbers that come out are something else, that means that, our model is wrong and we have to go and do something else. It's like we're trying to make the data good enough that we can disprove the model. We're trying to make the model simple and constrained enough that the model can be disproved, and all of these things kind of have to go together. So with that, I'll stop and point out the people that have done all this great stuff. So the first part of the talk was done by Matthew, who's now running his own lab in Edinburgh in Scotland. And the second part of the talk was like 90% of it was done by Xin Gao, postdoc currently in the lab. And then Mike is does all kinds of proteomics and helps with everything. I don't think there's a project in the lab now that isn't touched by Mike, so we just circle him in all the slides. And Mimi is here too. If you haven't seen Mimi yet in the conference, this is Mimi. And with that, I'll stop.
Thank you, Jan, for that talk. So if you have questions, please raise your hand and I will run towards you.
Well, I thought that was a great talk, and it explains something I've wondered about for a long time. One of the things that we've done in our laboratory is lots and lots of proteomics analysis, quantitative proteomics analysis across mammalian cells against dozens and dozens of microbial species. And what we do is we do something called a variance analysis. What are the proteins in a cell that never change? And part of this was to identify the standards we can use to calibrate the expression of other proteins too. And whether we went through mammalian cells or bacterial cells, the protein that always came out is always constant in copies per cell. Guess what? RNA polymerase II. So there is obviously very robust mechanisms to keep the protein levels in bacteria. Second thing is in microbes, as microbe growth goes up, the ribosomes go up much quicker than the mRNA level. So it was really interesting to see what happens in mammalian cells. So it's different sections.
The yeast are really similar. Yeah, that's right. Both the bacteria as well also scale up the mRNA.
Okay,
that's right.
Very nice. And so, do you see these processes of ribosomes and mRNAs and such as basically super modules that are probably composed of
I think they're
individual subunits?
Sensitive to some similar upstream signal. That's how I think about it. I mean, in a way, like the growth rate stuff has raised at least as many questions as we answered. And one of them is, how is that sensed at all? Like if it's eating trehalose or galactose or whatnot, why does it then regulate to make this many ribosomes? I don't really know. But the fact that both the ribosomes and the mRNA together go up like in the same kind of linear way makes me feel like, well, here that could be a common upstream signal, which is activating both of those biosynthetic processes. I mean, the cell can do whatever it wants, but that's our current hypotheses.
So that's maybe a follow-up question. Like there's two magical synchronization processes. One which synchronizes how much ribosomes and mRNA have, and the other one is the decay rate of all the mRNA is going down as you grow. Any idea what this could be, or those processes?
Yeah. So as the cell gets larger, I'll take the second question first. So as the cell gets larger, the mRNA decay rate is regulated in some way to decrease to compensate for the not quite linear increase in the transcription. We don't know what that mechanism is at the moment. That's Matthew is working on that. So I don't know what the answer is. I'm not working on it. The first question, but if you look across the different growth rates, not size, but just across the different growth rates, there the mRNA decay rate doesn't change very much. So in trehalose versus in glucose, let's say you have a 4 or 5x in the growth rate, the decay rate, the average decay rate for the mRNA is actually kind of similar. And that's really important reason, one of the underlying reasons why the concentration of mRNA is so much lower in trehalose compared to glucose. But not the only one. The synthesis rate also changes. Actually, like, could go through those slides with all the different parts of the model. And it's like, it was surprising to me, but you needed to know the decay rate. You needed to know the nuclear volume. You need to know actually the RNA polymerase concentration changes a little bit, but not too much, but you need that too, to really get the fit. Like, you put all those things in and then it works.
I have a very naive question. Would the age of the cell be considered as a factor in this increased growth rate?
Yeah. Well, so so we just did a study on yeast aging in collaboration with a group at Calico Labs in South San Francisco, and they've got these great devices to isolate, yeast populations of 20 divisions, 30 divisions, etc. And so you can get like a series of older and older yeast, which Mike then puts into the mass spectrometer. So then we can measure all of like the compositional changes as the yeast get older. One of the things that also happens as they get older, they get bigger. So budding yeast, because of the budding lifestyle, like it grows a little bit in G1, and then all of the growth goes into the daughter goes away. Then it grows a little bit in the next G1, produces another daughter. So each cell division cycle, it gets about 10% bigger. And long story short, I would say about 95 to 98% of the proteomic changes that are associated with aging are the same changes that you see when the cell just gets bigger and bigger. There are some important differences, but the vast majority of the changes associated with aging are actually deleterious effects in budding yeast of the cell having gotten too large. There's also, this isn't maybe what you asked, but this is what you're going to get anyway. There's some kind of deep link between a cell being senescent and it being too large, not just in yeast, but also, it seems, in human cells.
Any other questions? Oh, sorry.
Hi, this might be a stupid question, but given that your mRNA and ribosomes essentially scale with cell growth / volume, this would mean that dilution terms, when you model the system, essentially is not there. So what is actually stabilizing your proteins? Why wouldn't protein concentration just become unstable?
Yeah, so there's so many interesting aspects of your question. So I think in a proliferating cell, protein degradation to leading order is not relevant. So that the half-life of nearly all yeast proteins is longer than the cell division time. And that's also true in proliferating tissue culture, human cells, and all this kind of stuff. And so there, the relevant term is just dilution. So you have synthesis rate and then dilution because of growth, and then that's what you balance to get the concentration of the protein. I mean, a really interesting case is like, okay, what about all the other cells in our body which are terminal differentiated and just sitting there and not really growing? What happens then? How do they maintain protein concentrations? It has to be like a completely different process. Yeah, so that, I mean, this is only now starting to be measured with, different pulse labeling techniques and proteomics of human tissues and stuff like, or mouse tissues actually, and to get at some of those answers. But so the time scale of growth relative to the time scale of protein degradation is super important, as your question highlights. In the context I talk about here, we're in the one where dilution dominates, but that's not the only relevant case.
In the interest of time, let's thank the speaker again.